Good evening, class. My name is Peter Avalos, and this is my cell transport video. So what we have here is our lipid bilayer, and small and nonpolar molecules can easily pass through, such as my oxygen molecule here and my carbon dioxide molecule here. And that is due to the simple diffusion that no energy is required for them to pass through because they are small and nonpolar. And here we have our lipid bilayer with transport proteins, and that is because the glucose is a large and polar molecule, so it needs the membrane proteins to allow it to pass through the lipid bilayer. And this is moving from a high concentration to a low concentration. Here we have an example of active transport because ATP is being used by the sodium-potassium pump to pump sodium out of the cell as it pumps potassium into the cell. And this is against the concentration gradient, so therefore ATP is needed to be used to push out against the concentration gradients. And the potassium-sodium pump is found in the plasma membrane. Here we have endocytosis, which is the fusing of large molecules with the cell membrane, which will form vesicles which can be taken inside the cell. So step 1 will be the cell membrane surrounding the drops of fluid with dissolved solutes. Step 2 will be the cell membrane pinches in. And then step 3 will be the vesicle formation, and then it goes into the cytoplasm. Here we have exocytosis, which is basically the reverse of endocytosis. Step 1 will be the vesicle having products to be exported out of the cell. Step 2 will be the fusion of vesicle with plasma membrane. And then step 3 will be the expulsion of contents into the extracellular environment. And this can be done in bulk, and it releases the waste inside from the cell.